what I want to do today is continue a little bit the theme of yesterday, where we looked at how we can interact with the code and maybe show a few more things where you can do more than you could just through the graphical interface. I figured it's also good to look at a few apps just real quick, so you get an idea of what they're all about. And then sort of the second part of today, the idea is we're trying to pretend to do a real research problem. So we've done that often in class. It tends to work pretty well. We've not done it last year online, but figured it was missing, so we resurrected it. Hopefully it will work online. The idea is we're coming up with a question, we meaning you together with me, try to see if we can use one of the models, modify it, and answer the question. Of course, it's going to be very simplistic, but hopefully it will give you an idea how you could use that approach for a real research problem. And also, after this first session, we have a 45-minute session where we are discussing some topics, some questions that you sent. I think we have enough material to cover the 45 minutes, and we might run longer. And if you don't get to anything that you feel like we should have covered, just ping us again on Slack, and then we'll answer it there. Okay, so the first thing I want to do, actually we'll start here. So I just pulled up the app again, and I just want to quickly talk about some of the other ones so you know what they are. So the basic virus model, and the reason also why I'm talking about that is because I'll show you later an example of a research paper where they used something like this. This, we briefly glanced at it already yesterday, is another very simple model, but instead of a bacteria or some pathogen that divides by itself and lives outside of cells, this is an intracellular one. So you see what's modeled here is the uninfected cells, infected cells, and virus. And so earlier we had 2 equations, now we have 3, not much more complicated. But this is sort of the setup here. Uninfected cells get infected by virus. That leads to infected cells. These infected cells produce virus. That virus can go on to produce more uninfected cells. One thing our topic here is immunology, and there's no immune system here at all, right? It's just that process. So this is often called a target cell limited model. The reason for that is that these uninfected cells are often called the target cells, the cells the virus targets. And the reason why when you run it, you get an up and then a down is because the virus runs out of uninfected cells to infect, and then eventually it dies out. This is actually pretty much the SIR model on the population level in some sense, only that there's the virus explicitly. But it's the same idea. On the population level, you have uninfected hosts and infected hosts. And then, you sometimes model recovered ones. Here we don't model, the equivalent, which would not be recovered cells, but likely dead cells. But in either case, that compartment usually doesn't play a role. But you would get the same dynamics if you model an outbreak on the population level. Initially it grows, but then you run out of people that you can infect, and then it goes down. So that's the basic virus model. And here is the virus model exploration. It's the equivalent to the bacteria model exploration, where you scan over range, just a different underlying model. So, if you run it, figure looks very much like what we saw yesterday, only the underlying model that's run is now a different one. Then the virus immune response model is basically taking the basic virus model and throwing in several immune response components. So you see here, is the text large enough? I'll make it a bit larger. So you see here, uninfected cells, infected cells, virus. And then we also include a few immune response components. An innate response, very generic. So we don't split. We just think this is a totality, or it's some component that's important, like interferon or NK cells, or whatever you think is important for your system. And then T cells and B cells and antibodies. So you see it grew now from 3 to 7 equations. And of course, for each of those variables we track, we have to specify processes, mechanisms. That's the whole point of our models. And so you see things get a bit messier. The model gets bigger, more equations. And of course, you have to give numbers to all those equations. So this is already getting a bit more complex to run and to explore. And also, of course, if you were to apply that to any real pathogen, you would have to get parameter values from the literature for all these parameters that match what you're trying to study. And so, if you run it, this looks bad because something goes through the roof. You see a lot of things up and down, so there's lots to explore in this one. Then here, these I just mentioned, the antiviral treatment model is just basically the virus model again. But now what you have here is these additional terms that are here in green that mimic a drug. So the idea is the drug could either reduce the production of virions, that would be acting on this term, or it could reduce the rate at which cells get infected, that would be at this term. And we think of it here in terms of these are two hypotheses, and you are using your model, and in this case, actually comparing it to data. You're not fitting it to data here rigorously, but there is data, and that you compare it to. And so here you can see this actually from a paper, and in that paper, they actually fit the model. But here we can look at it. So this is several patients, and you see that kind of kinetics, and then you basically run your model, either with the drug doing one thing or the drug doing another thing, and then you can compare, and that might tell you, okay, it's more likely that the drug works one way or the drug works another way. So that's getting, this is not just exploring the model anymore, how it does. This is starting to, because you have data here, this is starting to do some kind of testing, comparing, maybe ruling out certain models, finding more support for other models. And in our case, because every model has mechanisms, once you have sort of support for a certain model, that also means the mechanisms that you implemented. Of course, you can never show that a model is right, but if you find some models that are built based on what you think is going on and they don't match the data, you've learned something. And then you can sort of keep progressing, and the more data you get and the more insight you have, the more fine-tuning you can do with your model, and the more likely you get to a model that you think is pretty reliable, and then maybe at some point you can use it for prediction. The influenza drug model is somewhat similar. It's again the same model as you saw before. The difference now here is that it's actually being fit to data. I think I should have changed it to a log scale. I don't know. I want to do it. Somehow it froze on me. Okay, we'll see. Hopefully it will recover in a second. And I didn't crash the server. But basically, this is the first one where you do some fitting here. And then fitting is not much explained here, but it's further explained than down here in these other models. Looking at it again. Okay, so here you see it's a bit busy because you have different data sets. It's from a study where they gave the drug early or late or not at all. And so then you're trying to fit that and see, which of these models matches the data best. So this is actual, statistical fitting, while this one we might have called as, fitting by eye. You look qualitatively if you can get agreement. Sometimes the differences or similarities between the data and your models are striking that you don't even need statistics to know that this isn't working. If you have pretty reasonable models that get close to the data and you can't really distinguish by eye, then might have to use more rigorous statistical measures. And that's covered here in the model comparison app. So that's one where you try two models and use something called AIC, which is a measure that tries to discriminate between model fit quality. And this one covers the idea of confidence intervals. And then here we're going to spend a little bit of time on those. Here gives you, and we had this question before, is, how do you know how to build your model? First, of course, you decide, do I want one of these compartmental models? Do I want an ODE model? But even with that, there are many ways you can implement the models. And that's really something that, unfortunately, you can't thoroughly explore. You can maybe try a few model variants that seem reasonable. But basically what this shows is you have a not too complicated model in terms of variables. There's just an innate and adaptive, but there's no multiple ways in which the innate and the adaptive response can do their thing. So either proportional to virus or proportional to virus in infected cells. And they're all somewhat reasonable. Of course, every model you want to build should be based on some reasonable assumptions, but quite often we still don't know the immunology, the virology enough to know it's really this mechanism or that. And so this app lets you explore what changes and doesn't change if you assume the process is like this or like that. So that's always something you need to keep in mind when you build your models and also when you read the literature. They implemented it in one way that's hopefully reasonable, but there's probably alternative reasonable ways you could have implemented the model based on what you know about the system, and it might or might not lead to similar results. That's just an inherent nature of the way this works. You can explore it somewhat, but you can never explore it fully. And of course, this is not just models, right? I mean, if you're an experimentalist and you run an experiment in B6 mice, you could test if it also works in bulb C mice, and then you could try, if you had a lot of money, 10 different other mouse strains, but you'll never be exhaustive. And in the end, we always have models. These models could be computational models or they could be experimental models. And we hope that what we learn from the models has some applicability to the real thing, whatever the real thing is. But in the end, it's always tentative until you're able to really confirm it or test it in the real system. For a lot of things, eventually for a drug or for a vaccine, we have to go into humans. But hopefully we gather enough information and understanding in some other system that we can use that when we go to the real system. So that's the model variant. I'm not going to talk much about that. And then uncertainty and sensitivity analysis, we'll get to it a bit. So that's one way where you can get, earlier we said, maybe we know our parameters not always exactly. So if you have data, you can try to estimate it. Otherwise, you go to the literature and find it. You can also do the thing where you scan over a parameter, but you can only do this many at a time. If you have a bunch of parameters and you all know them somewhat well, but not too well, you can use uncertainty and sensitivity analysis, and we'll play with that a little bit. And then, of course, another source where you can get randomness is when you run a stochastic model. So this one is, again, the same basic virus model, and this is how it would look like as an ODE, but then you could implement it as a stochastic model. And then when you do that, you see every time something else might happen. In fact, you see here that a few times when you run it, you don't get an outbreak at all, meaning the infection doesn't take off. Other times it does take off. This you can only get from a stochastic model. For a deterministic model, once you have your parameters, you run it, you always get the same result. That's not the case here. So stochastic models are harder to deal with because instead of just running it once, you have to run it multiple times. They're also still much harder to fit to data, but they're in some sense more realistic because every real system has a certain amount of stochasticity. So you have to decide if the question you have requires one of these models, and then you might want to use it, or you think you can get away with the ODE model. And most of the time when the numbers are large, you see here, it looks pretty smooth because once the numbers are pretty large, then the deterministic ODE one is usually a good approximation. When the numbers are low, then you might have to look at stochastic models. Okay, so that was a quick run through the different apps, so you just get a bit of a bearing of what they're all about and what they do. And we'll look at a couple in a bit more detail. So there's a question, can we always use AIC? What can we use when not all the models use maximum likelihood? For these kind of models, you always have a goodness of fit that you can probably formulate as a maximum likelihood. So you should always have that. Now, if you have enough data, my preferred approach is to actually use cross-validation or a train test approach. Like you fit your model on some part of the data, and then you see how well it works on the other part of the data. Because in the end, for every model, you don't really want the model to be perfect at explaining your specific data set. You don't want it to sort of capture every little wiggle and piece of noise in your data. You want it to capture the general pattern of a system like that. Meaning if you redid the experiment, you would likely get slightly different jitter, but you want to capture the overall behavior. And things like AIC are trying to do that, sort of make a best guess of what model might be best if you applied it to future data. I think something like cross-validation or sampling is even better. The problem is if you don't have a lot of data, then that might not work very well. But if you have enough data, then I think that's an approach I would recommend. Okay, let me know if you have any other questions on either the whole thing or what I just talked about. You just unmute or type. Okay, then what I want to do now is I'll show a little bit more how you can get to the apps with code, and to speed up things a bit. I've started coding it already, so I don't have to look for the commands that I sometimes don't remember. I'll give you those scripts again, like the one from yesterday. So here's going back to the basic bacteria model exploration. We looked at that again. So first we load the package, and then we run it here with the default values. And we saw yesterday that this one does not return a time series. It underlying runs the model, and the model produces a time series, but then it extracts just a peak and the final one from the time series and does it for multiple parameters. So when we look at the output, you see instead of having here time and B and I, like we saw for the underlying model, it now just returns the peak and the steady state for each one of the sample parameters. In this case, we're sampling parameter G, and we're producing 10 samples. And so we made this little plot here yesterday, which doesn't look as pretty as the one through the graphical interface, but it's basically the same. You scan over one and you look at one of the outcomes. Now I want to show you an example here. Let's say you wanted to actually explore 2 parameters. Not just G, but you want to see how combinations of G and some other parameter influence it. In principle, you can do that for an infinite number of parameters, but at some point it gets really slow and really unwieldy, and it's also hard to show into plots. So normally people only do, scan over 2 parameters and maybe then do a few values of a third or something like that. Usually, naturally, you have some parameters that you're more interested in because they match your scientific questions, and other parameters you're less interested in. So you'd focus on the ones you're more interested in scanning, and the other ones you might either keep completely fixed, or you might do something like sample them with uncertainty analysis, as we'll look at in a minute. So what I'm doing here is I'm using that simulate basic bacterial model exploration, which loops over 1 parameter, and then I manually write another loop around that changes another parameter. So in this case, I decided I'm going to change the parameter I. So I make a vector of values for the parameter, or DI, sorry, the death rate of the immune response, the decay rate. I'm also deciding here, the samples, instead of hard coding them in there, I give them a variable. That means I can more easily change it here. So that's always a good thing, to not sort of hard code numbers, but to specify it somewhere and then just use that. So I say I want 20 samples for G for each of 10 samples of I. And then here I'm just setting up, for simplicity, I'm saying I'm only interested in the bacteria peak, but of course I could be interested in all 4 of the outcomes. In fact, why don't we just do that quickly? Let's say I'm also interested in the peak of I. So I'm just setting up 2 arrays that are just filled with 0, so I can store my values. And so here, this is the main thing. I'm now looping over every value of this DI vector. And you see here, instead of having a fixed DI like I had up there, I now have a different value every time. And for every value of that time, I underlying still run this thing here, sampling over the parameter G from 2 to 10. And right now I take this many samples, which I set to 20. So basically I now do 10 different DI values. And for every DI value, I run 20 samples of the parameter G. So that's sort of going along a grid, basically. So it will take some time. I wanted to also know now I, so let's store that here too. So then every time I run that, I basically pull out just this B peak entry, these 10 values, and stick it in my array here. It's basically a matrix, sort of every column I go, every row I go down, and the row is those guys. And then for each one, I put in the 20 entries I get here from this one here. And so then I end up with the structure that's filled that has 20 rows, no, 20 columns and 10 rows right now. So you can actually see that up here and in the environment output, you see the structure, 10 rows, 20 columns. And now you see they've gotten all filled with numbers. And so, earlier I would make these kind of plots. Now what I can do for 2 parameters at a time, I could, for instance, make some kind of heat map. So you see, this is now a not very good looking one, but just, very basic heat map where, I go over one and then I go over the other. Let me see. So if I might want to define the axis, this is the IVAC, and this, I guess, is I need to recreate this thing here, which is the G vector. Let's see if I did it right. Did I switch them around? Yeah. Do you think it's the other way around? Okay, so now you saw earlier it was just 0 to 1 on these axes, which is not that good. Now it's actually the values. So these are the values of the I vector, which has varied from 1 to 10. And these are the values of the G vector, which I did from 2 to 10. I guess I could do 2 to 20, so it looks different. And then we could make the same for I peak, and look at that. Of course, the figure doesn't look that great, but I hope you get the idea that you can here loop over 2 parameters at a time, and in principle you could wrap another loop around it to have a grid of 3. Of course, the computational time multiplies because you always have to, for every outer loop, you have to run stuff in the inner loop. So those are the 2 now. And also visually, you can do a heat map here. After that, it's getting hard. But those are the kind of figures you might see in papers as well. So, this earlier figure is the kind you'd probably often see in papers where you have something along the x-axis and some outcome along the y-axis. And then, some version of those kind of figures. Those are very common for papers that use models to explore outcomes. So that's what I wanted to show you here, that idea you can apply a loop. And of course, you could do that too. You could, there is no exploration app for the virus and immune response model. I just did it for those 2. But you could now basically make your own, right? You could make a loop, and then you can say, okay, I'm calling underneath this model here, and I'm looping over one of these parameters of this model, or 2. So then you would have basically built your own basic exploration model by just using the underlying simulator and writing a little bit of code around it to explore it. Okay, that's what I wanted to do still on this one. Any questions or comments on this? Okay, now at this point, I'm not sure if silence means it's totally confusing or it's totally clear. I have to assume it's clear enough that I move on, so I'll just do that. But do keep asking if there's anything confusing or unclear. There's a question, can I show a legend for the heat map? Yes, in principle, you can do that. I don't remember the command right now. Maybe there's a legend command. Again, if you do that for real, I guess you can somehow do this. Let's see if there's a quick. Okay. I don't know. But actually, what I would recommend is, if you made a real figure like that, again, use probably ggplot based and packages that tie into ggplot, you can get much better looking, nicer images quicker. So I would probably not use base plot commands to make that. This was just the first command that I came across, and then I don't have to much reformat the data. But again, ggplot is so popular and so many people use it that whatever your question is, you just need to Google it and say, heat map plot, ggplot R or something, and you'll find someone showing you how to do it. So that's how I do it, because I can never really remember those things. So, but the good thing is you really quickly find someone who has an example that's close enough to what you need. You just switch out their data with yours and do it. So, and if I can share the script, yeah, I'll do that. I'll post it on the SMI webpage again. So yeah, if you have specific figures, often the main tricky bit is deciding which way do you want to present your data. First it's table or figure. I'm a fan of figures, but sometimes tables are good too. And then once you've decided that, the question is, how do I show it such that I can communicate the best way possible, what I'm trying to communicate here. And that can be line plots, can be 2D. People generally don't like 3D plots because they tend to be more confusing than enlightening. So I would most of the time stay away from those. But there are all kinds of charts. We'll see now an example of a box plot. You can have all kinds of types of plots. So, often you can just look around, and if you know kind of the structure of your data, then you can look what kind of figures are people making and decide, oh, this is a good one. That would illustrate it very well. Of course, here you also have to make choices about putting something on the log scale, either one of these or not. So there's lots of different options here. And Cody put an example in the link from Stack Overflow. That's where I always get my examples from if I need to figure out what to do. Really, the hard bit is to know what to Google for. Once you know what to search, it's easy to find it. You just need to know what that thing is called that you want to do. Okay, now I want to switch to the sensitivity and uncertainty analysis just very briefly. So the idea here is, as I mentioned, there's almost always you don't know your parameters exactly. And especially if you have a model where you not just want to sort of explore a general model for, let's say, an acute virus infection or a general model for a bacteria infection, but a very specific system. You'd say, I'm trying to model flu or something. Then you, of course, want to have parameters in your model that correspond to the real system. The lifespan of the cell, the growth rate of the virus, the rate at which the cell produces virus, all these kind of things. Sometimes people just decide, okay, this is the value that I'm going with, and keep it fixed. That's of course never really true, right? Even if we had precise measurements, we know if the temperature in the room is a little bit higher or lower, things might change. So almost always there's a range. And so one way we've already looked at is the way you can deal with ranges is what we just saw here, right? You can scan systematically over it and see, okay, if I change that value from here to here to here, how does the outcome that I'm interested in change? You can do that if you have a few parameters or some that you're very interested, but if you have a somewhat big model with a lot of parameters, then that gets kind of hard to do. You would have to make a ton of different loops. Also, sometimes you might not really be interested in that parameter too much. You just, let's say we're really interested in that parameter here, the death rate of the infected cells. We're not that much interested in that growth rate parameter here, but we just don't know it very exactly. So we need to account for the fact that we don't know it very well, but we don't really want to scan over it in detail. And then an approach you can use is you can basically sample over parameters. So instead of now taking a fixed value for a parameter, you choose a range. And the distribution could be anything. Quite often uniform is fine because you really don't know it that much anyway. So you give it an upper and a lower bound. But you can also do other distributions. You can say, I know this parameter is normally distributed with a mean of this and some standard deviation. So different ways you can do it. And in this example, most of them are just uniform. That's sort of the easiest one, but just for sake of illustration, I had here the mean different. And I think it's actually implemented as a log normal, because one thing you need to be careful when you do sampling, you don't want to sample parameters that make no sense. And pretty much all the time here, all your parameters are positive. It's rare that you build a model where your parameter is negative. I mean, you could, but usually it's set up such that parameters are considered positive. And so you want to make sure you don't accidentally sample a negative value. So, if you draw from a normal distribution, that's unbounded. It goes from minus infinity to plus infinity. So if you draw from something else like a log normal or a gamma, then you can prevent that problem. So what you do then is, instead of picking a fixed parameter, you say, okay, I'm drawing 100 samples of parameter. And for each sample, you sample from every parameter. You just have some underlying routine that grabs a value out of that range that you specified. And then you run the model for that. And then instead of now getting a single result like you did earlier for, B peak or I peak, you now get a distribution because you have different parameter values. And while our models are deterministic, meaning you get always the same result for the same inputs, now here you're changing your inputs. You have different parameter values, so the outcome is different. So this app lets you explore this. Here's an example. Just run it with the default. We're looking at the bacteria model again, and we're assuming that the outcomes we're interested in is B peak, B steady, and I steady. Just those three. I just picked those. But if you're doing it, you can decide based on, what your, system of interest is and the outcome of interest. Those are the ones you would focus on. But the setup is the same. You would run your model, you would pull out that value, you would change the parameter values, you would run the model again, you would pull out your values, and so on. And then in the end, instead of just a single number, as we would have gotten earlier, you now get a distribution. So again, underlying, you run your time series, but then the time series is just used to extract what you need and then plot it. And so then you get these kind of box plots. So now you get a measure of uncertainty. And that's generally good to, have not just point estimates or point values, but uncertainties. And so basically what you're saying here is, now, given these ranges for the parameters, this is what I would expect the outcome to be for B steady. So instead of just a single one, you get the spread now. And you can combine that with this approach. You could say, well, I'm writing some code where I'm looping over one of them. And then while I'm looping over this one, write a little bit of code like this. Sorry, where is it? Here we go. A little bit of code like that loops. At the same time, you can then do the sample for the others. And we can actually try that. So here, this is our graphical interface. And here again, I'm now just looking at what the underlying function is. As always, you go to further information. It will tell you that the function that's running under the hood is called simulate US analysis. And that itself calls the bacteria ODE model that's run. So then I could just, instead of calling it this way, I can now just call it here with code. And again, it takes a little bit because I'm running it now for 10 samples, 10 times. So you see it's similar to earlier, but now instead of systematically varying, it's just randomly pulling out values between those ranges. And because it's random, you also have to sort of want to be reproducible. So for that, you set a random number seed. And that means you pick the same seed, you always get the same random numbers. Random numbers in the computer are never really random. They're pseudo-random, which allows you to reproduce the same thing. And you look, here's the outcome now. Again, not the time series, but from the time series, the simulation function reads off these values here. These are the outcomes, peak, steady, steady, and these are the inputs. So you're seeing we're sampling them all of there, including now the initial conditions. You can do that. You know, we kind of call the parameters, you know, parameters, and the things for the variables, the starting values, initial conditions. But for this kind of purpose, you can think of them all as parameters or inputs that can be sampled. In general, these starting values have less of an impact on outcome as the parent model parameters. So this is, you know, now you can make a box plot by, you know, just looking at, instead of a single value, you just have a distribution here, and now this gives you a box plot. Now we just ran it for 10 samples. That's of course not a lot. It just made it so it goes faster. But in reality, we'd probably want to do it for more. So that's one part. This gives you uncertainty, sort of given the input, given the variation in all these inputs, what's the variation in the output? I'll get to the questions in a second. I'll see you, and I see it in the chat. So just, I'll get to it. And the second one, you can then go one step further and say, okay, given that I vary this parameter, how does the outcome vary with that parameter, while at the same time all the other parameters vary? So it's a little bit like we saw earlier, where you scanned along a parameter. Just now, instead of systematically you choose the values and scan it, you just have sampled values, and the other parameters vary at the same time. And in fact, you get that here. You see at the same time here, we get, you know, values for the outcome, we have values for the parameter. So we don't actually have to run the code again. We could actually plot it right away. Let me show you that so you're convinced that this is, can take the same outcome. So this now shows more specifically, as G is randomly varied, sampled, and the other ones are sampled at the same time, how does it affect the outcome? So here we only have 10 samples, so it's a little bit hard to see. So I have it here run again for 100 samples. That will take just a little bit longer. And then you will see that it looks a bit, you know, more systematic, or you can see the pattern better. And remember back that we can actually write down the equation for the steady state here. So in this case, we wouldn't actually need to do this sampling because we know exactly how steady state depends on G. But most of the time, your model is so complicated that you can't write down the math for this, or the outcome you're interested in just doesn't allow you to do the math for it. And then you would get it from here. So we see here that, you know, there's pretty much a linear relation here. The reason why it's not completely linear is that, remember, as G varies, all the other parameters are varied at the same time. And they, of course, can impact the outcome too. So that's why you get this scatter, the sample. And so technically, the first part, the box plots, is called the uncertainty analysis, because we're looking just at how uncertain is the outcome, given the uncertainty in the inputs. And this part here is called the sensitivity analysis. It's how sensitive is the outcome to changes in a specific parameter. Of course, we could make these plots now for any parameter outcome pair we have. You know, you can see we have here 3 outcomes and 2, 4, 6, 8 parameters. So that gives you 24 plots. So it's, you know, it can get messy. Usually in a paper, you don't want to show all these scatter plots, but it's still a good idea for yourself to just look at them and look at the patterns. So here we could, this is now B steady, and you see it basically doesn't really depend on G. And again, we know that from the equation that B steady does actually not depend on G. And then you can also compute correlation coefficients for those to sort of summarize the whole plot, and that's shown there. Okay, now let me pause here real quick and get to the question, what is the parameter for scatter plot? Well, I'm not sure I understand. Maybe it's in here in the app. So here you pick whichever one you want to vary. If you produce a scatter plot. If you do a box plot, that doesn't matter. But if you quit to having a scatter plot, then here you just should decide which one you want to see on the x-axis, which is basically equivalent to the code here where I said, I want G here on the x-axis. That's my x-axis. So that's just for what you want to see. It's, underneath you always run the same thing, then you just look at different plots here. Now, again, this thing is set up that you have to run the model every time you want to see the different plots. The good thing is when you do it here, you don't. We can just run the model once. And that's good because in real life, you would probably want to run that for a lot of samples, 1,000 or 10,000 samples, which will take a while. But once you have it once, you can just do all the plots you want. So this would be the one for B. Again, showing that the steady state does not depend on the initial condition for B, how we start B. I hope I answered that question. If not, post a follow-up one. And there's a hand raised.
Yeah, hi. Sorry if I missed that. Like I was thinking about seed. So I read somewhere like seed is something that allows us to get like similar results if we run the model several times. So that's what seed does, that we can give some number to seed and it gives like some starting number of a large set or run.
Yes, that's right. So basically you can think of these pseudo-random numbers as just a sequence of random numbers, but the sequence is always the same. So if you always start at position 100, then the following infinite number of sequence of random numbers will always be the same. So you basically just sort of start at a different place in that sequence, and then all the numbers that follow are in some sense random because, it's, you don't know what comes next, but they're computed by an algorithm, which just always computes the next number. So you actually know what you're getting. So basically the way you look at, are these random numbers actually random, is, you would draw a lot and then you would plot them and make sure they're, nicely spread out and things like that. The good thing is nowadays all software packages have random number generators that are pretty robust. That used to not be the case 20 years ago. I remember when I learned about them, we were told to be careful because there are some random number generators that don't actually produce real random numbers. And so results might not be fully reliable. But nowadays there are, and also for these purposes here, I think it's perfectly fine what you're getting there. But yeah, so you want to, because when you change something here now, let's say I changed something in the inputs and I ran it again, I want to make sure that the change I'm seeing is just due to what I changed and not just because I got another random sequence. Otherwise, debugging your code would be really hard. So that's why you want to have, in some sense, reproducibility. So you have a random seed there. And then if you don't want a random seed here, you can, instead of having some hard-coded number, you can just feed it something like the system time. And the system time changes every time you run it. So every time you would get a different result. This is how you could make it truly random, but then it's not reproducible anymore, unless you remembered what time exactly you ran the code. Other questions?
If there is no other question, like would it be possible just a quick example how we can we can replace this 100 with some time sequence or something like that?
I think the command is sys.time. We do turn it into some number. So see, that gives me the system time. And I could shove it right in there, but I'm doing it separately. So no, I can. So if I run it with this one, with the 100, I always get the same one. If I run it with this, I always get something different. Maybe I should do fewer samples. Just illustrate real quick. I'm going to run it now as soon as this is done here. Let me run it first again with the fixed seed twice. I'm going to run it. And we make the plot. So we run it again with the same seed. I make the same plot again, it shouldn't change. It was the same plot. So that's the fixed seed. Now we're going to use the system time as the seed. So we run it once. Make a plot. Now we run it again, picking a new system time because a few seconds have changed. Run it again. And now the plot should not be the same. It should look different. See, it changed. So that's how you can, for instance, very quickly give a truly random one. In general, I would not recommend doing this. I would most of the time stick with something that, is reproducible. So, 3 weeks later, if you're asked to, change that figure and you don't want to have a completely different figure just because, your random number seed changed. If you do it this way and all they want is to have the, the points to be blue instead of red or something. And if you didn't pick a fixed seed, then your whole figure will change, potentially. If you have a fixed seed, you can just reproduce it again. So in general, it's a good idea to have fixed seeds. Other questions? Yeah, that's Cody wrote in the chat. You can print it and you could save the system time, in your workspace. But I'm not sure what it helps, what it does, other than the advantage of setting a fixed seed. But sometimes you might want this. Okay, we have a few more minutes. Why don't I show an example that I hadn't planned? So we'll see if it works. Just showing how you could combine this one from the earlier, this systematic scanning here with the randomness. So I'm copying this part over here. I'll clean it up later. For now, I'm just keeping it messy here. But now instead of calling the basic bacteria simulation one in here, I'm gonna call this one here. So now what I'm trying to do here is I want to have some parameter that I say I'm really interested in. Let's say this is my DI here. And the other ones I want to sample because I'm not that interested in them. Now the way I could do that is that I just set for my DI vec, the minimum and the maximum, I'm choosing the same and exactly the value I want. This basically forces it to not sample it, right? It will draw 10 samples, but because the minimum and the max is the same, the samples it draws will always be exactly that number, right? So I basically artificially force it to not have variability there, but instead it gets me what I want here. The rest is being sampled. And then I can still have B peak here. So that should work. Just keep calling it different things. And I also want to do this. So what I'm doing here, I'm making a loop over this DI, and I'm giving it specific values, 10 values from 1 to 10. And then for each one of those DIs, which are basically forced to be fixed here, I'll sample over the rest of the parameters. This many samples, P samples, again, say I'm telling it to do 20. Then it runs it for 20 samples. It gives me this thing here back. I'm pulling out the samples. So now what I should get, instead of sort of, in the end, a single box plot, I should now be able to do a box plot for every value of DI. So let's see if my code runs or not. It might take a while. In the meantime, I can think of how to do it. I'm not sure if box plot can. We'll see. So now I have this B peak all. Now I would like to have 10 box plots for each one. I don't remember if feeding it the whole thing is a. I got it. It's just not. It's just now the other way around. So not quite what I want. So that is easy to change. The command T transforms it. So basically it flips it around. And now I got what I wanted. So, compare that with what we had here earlier. Of course, the plot is long gone, but I guess we can resurrect it. That was a single one, right? We changed something. In this case, we changed G here, right? I should compare it to DI, but you get the idea. You change one across, and then for each one, you get a single answer. What we're doing here is we are changing something along the x-axis. In this case, I changed DI. And then, but for each one, then I sample all the other parameters. And I get therefore now distributions of the outcome instead of just a single one. So that's to some extent a better way of, exploring your model instead of saying, okay, I know them all accurately and I just sample over this. Here you're saying, okay, I'm really interested in this one here, and I go over it, and then I look at the distribution of the others. You might see here that there's actually some at 0, right? And so basically what that means, there must be some combinations of parameters where the bacteria are so wimpy that they never take off. And just, there's nothing happening. And so there's no real peak there. They're just declining. So you might not want that, right? That's one thing you need to be cautious about. And when you work through the app, one of the tasks is sort of telling you how to do that or explain it. You want to be careful that you're not basically having two very distinct regimes that you're combining together in a box plot. One regime where the outbreak happened and one where not. I mean, you can make a box plot that spreads over all those outcomes, but it's maybe not meaningful. Maybe you should focus on sampling a range where you always get an outbreak and you're just looking at the size, and then another one where you don't. Or you sample over the whole, but then you separate and you say, out of my 100 samples, 50 times I actually got a chronic infection with bacteria and immune response at the end, and 50 times I didn't. And then of the 50 times where you get it, there you might then compute a mean or median, but you might not want to compute means or medians or box plots over the whole thing. And of course, conceptually now we could, look at the scatter plot for each value of what we scanned along. For each value of DI, we could make a scatter plot instead of a box plot and look how things changed for some other parameter. Of course, not DI because we kept that fixed, but we can say for every value of DI, let's make a scatter plot of this outcome versus R or something like that. So you can start, exploring in a good bit of detail, and you can always dig deeper. At some point, you just decide, okay, this I've explored it now. This is what I want to convey here, and then stop at that point. So I hope I gave you a bit of an idea how you can potentially use these underlying simulation functions with some code wrapped around to take you a bit further, and especially if you explore and learn. Again, for research purposes, and we'll do that later now, you might likely not be happy with just having this bacteria model underneath. You want a different model. So then you will have to actually hack the code. But you can get pretty far, at least in terms of learning and understanding, by using it this way.